I'd like to introduce Dr. Phil Tarr, who's a professor of pediatrics and the director of gastroenterology, hepatology, and nutrition at Washington University in St. Louis. And his work has most recently really been involved in understanding a very specific organ system and the relationship of the microbiome and fetal health. So thank you, Phil.
Thank you, Bonnie. Let's see. Okay. Very good. So I really appreciate the opportunity to come today to talk to this Child X Symposium for multiple reasons. First, I'm going to be talking about necrotizing enterocolitis, an utterly horrible and inexcusably overlooked disorder of early childhood. I also want to convey that we are at a point now where technological advances are giving some clarity to the etiology of this disorder, and it will be technological advances looking forward that may help us use this knowledge to prevent this terrible outcome. An additional item that I'd like to convey is I think that we are now at a point, as suggested by David, and I must say that his talk and mine, without any rehearsal, have converged on very similar observations and recommendations. But I think we're now at a point where we can start to use microbiome science for prediction of outcomes, that we're getting beyond an era of cross-sectional comparisons, and this will, we hope, greatly reward the NIH and others for investing in this new frontier of biology. So I want to thank Bonnie and everybody for this invitation. I also want to call in advance the ecological team that has really brought us to this point, and in particular, Dr. Barb Warner, a neonatologist whose skills have been incredible in generating much of the data that I'll be talking about today. So, necrotizing enterocolitis. Anyone in this room who's trained in pediatrics will have recognized this story. A 26-week gestational age pregnancy ended rather abruptly, producing a boy weighing 725 grams. To put that in perspective, that's about the weight of 2 Coca-Cola cans. Neonatologists have gotten very good at keeping these children alive. They've largely done it by their expertise in disorders above the diaphragm, with major advances in pulmonary survival. Nonetheless, this child, like so many children worldwide who are born at this gestational age, was given a lot of antibiotics at birth, largely empirically, and this child's first antimicrobial day of life was actually day of life 18. None of the indications for giving this child antibiotics were what we call incredibly high grade. The mother's pathology report from the placenta was, quote, chorioamnionitis, which is a nebulous diagnosis. When the child didn't get off the ventilator in the first week of life, and by the way, neonatologists are getting children off, if they're even putting them on ventilators, they're getting them off the ventilator within a couple days, which to those of us who've trained several decades ago is incomprehensible. This is fantastic. This child took about a week to get off, and everyone was worried something was going on. And the child had a persistently high white count. Nonetheless, the child, after a couple failed attempts at extubation, seemed to be permanently extubated at the day of life 20. So, child's now beyond the first 2-week mortality zone.
Excuse me, we seem to have a mic problem.
Oh.
I'm going to allow you to do that.
Oh, dear.
It's on. You can just speak into it normally.
Okay. The old-fashioned way. So this child was extubated day of life 20, has survived the hazardous first 2 weeks of life, and now at this point everyone can more or less hope for the best. Until about a week later, the child in the middle of the night produced 1 bloody stool, Severe abdominal distension. This happened well within the course of an hour. Abdominal x-ray demonstrates a lot of air in the bowel. Nasogastric tube was placed. The surgeons were called. Antibiotics were again started. The child was reintubated. An x-ray was taken again a little after 8 in the morning. You can see a rim of air over to the right side of the abdomen there. This is called pneumatosis. It's gas in the wall of the bowel. It shouldn't be there. It's a highly ominous sign. The patient went to a laparotomy. This is what the bowel looked like. The surgeon mercifully didn't attempt to be a hero, said this is not amenable to an operation, and the child died several hours later. This child had a disorder called necrotizing enterocolitis. It's an acute inflammatory injury. It's not rare. About 5 to 10% of children born under 1,500 grams worldwide, decade after decade, will sustain grade 2 or grade 3 necrotizing enterocolitis. It is 30 to 40% fatal. The only difference between this and an anterior wall myocardial infarction in 2016 is that this disorder is about 10 times more lethal. Among the survivors, the 2/3 of the patients who survive, some do well, but many do not. It is an independent and fairly strong risk factor for later in life neurodevelopmental issues. The treatment is very crude and has not changed in 4 decades. Our ability to detect this disorder in advance of abdominal distension and clinical deterioration has not changed in this interval either. Effectively, the surgeons do a laparotomy when indicated in about half the cases and see if there's anything to resect or not. If one were to try to approach necrotizing enterocolitis to figure out what is causing this disaster, go to a review article, and here are the suspects that have been variably put together over the years, and there's certainly a microbial flavor to this. Prematurity is an enduring risk factor for the development of necrotizing enterocolitis, and I think we can more or less assume that a preterm infant is going to have a different gut microbial population than a term infant in whom this disorder rarely develops. Formula feeds have been associated, though regrettably, despite the best attempts, the use of breast milk has not made this disease go away. H2 blockers, acid suppression is a risk factor. These are all, you can imagine, large database analyses. And prolonged antibiotics in most, but not all studies, these are prolonged antibiotics at birth, beyond the 5 days, are associated with the development of necrotizing enterocolitis over the next couple months. So with this, several years ago, in response to a demonstration project request for applications from the NIH, we entered a proposal to test the hypothesis that bacterial dysbiosis, defined as a community of microbes that's associated with an unfavorable outcome, precedes and thereby plausibly causes necrotizing enterocolitis. One of the concepts David just mentioned was if you're trying to associate something in an epidemiologic study and something that you are trying to put forward as causative, it's better to be there before the event than only at the event. Now, we have a lot of opportunities in looking at necrotizing enterocolitis. These children live in a managed ecosphere, not quite as well managed as Dr. Flakes Lamb's from Philadelphia, but these children are in incubators. The nurses do not wear gloves when they touch them. The parents do not wear gloves, but all other visitors wear gloves. This disorder occurs days of life 14 to 60. You don't have to wait several decades to see if somebody will become obese. So you have a horizon. You can reasonably anticipate an outcome, a little shorter than pregnancy, but it's not so unfeasible that, so it's exceptionally feasible to look at this, to look at biologics prior to the event and see if they are related to an event in a reasonably short period of time. The 5 to 10% attack rate is fairly appreciable. It's not 1 in 1,000. These children live in this venue for 2 to 4 months. They're not running around the community, and you have to pull them back for specimens. There's extensive metadata, one of the very good things about electronic medical records. If something happened, was documented, you can retrieve it for years to come. And the specimen of interest, stool, is a relatively low-risk material to take possession of. but there are challenges. There is certainly the concern that this is an observational study. There were really no promising leads on which to build any highly robust hypotheses at the beginning of this disorder. And our biggest challenge was the normal. In any case control study, you have to have a normal, was not defined in this ecological sphere. So this is what happens at birth. The child is born, the bowel is full of meconium, and as we've just heard, it's either sterile or, quote, largely sterile. It's certainly not a vibrant, replicating bacterial community in the meconium, or we would have discovered that years ago. And over the next hours to days, the meconium passes, and soon a variety of bacteria come in. There's gram-positive cocci, there's gram-negative rods. This is my rendition of the bacteria that are now going to be with you forever. There may be some turnover, there may be changes in proportion, but you will never have a sterile gut again after the first few days of life, and it is a fantastic opportunity to study the ecology of the in-migration of a population that we all thought was, that we still think is largely sterile. Others have tried to define this. For over a century, we relied on culture technology, and we all know that that has some value, but an awful lot of expense and may be inaccurate. We then went through a couple decades of gel-based PCR-derived work, where one gets patterns and barcodes, and that's pretty good for identifying diversity changes, but it is not very good for really enumerating the bacteria in a stool. It's very expensive to try to extract data from that technology. And it wasn't until about a decade ago, following the pioneering efforts of Dr. Relman, that we were able to go directly to this biomass and amplify from the stool and do a census to determine who's there. We chose to use 16S ribosomal RNA sequencing at the time. We began the study in the late, as we approached 2010. This exploits the ribosomal RNA gene of bacteria, extract DNA from stool, prime it in one of multiple places, sequence what between your primers directly from your amplicons, send it into a machine, and you will be able to get a number of reads to tell you who's there, what are the bacteria that are there. We use 454 sequencing technology. We had 6,000 to 7,000 reads per sample, and this was all referred to the ribosomal database. And then we started to take a few children who developed NAC and a few children who were controls, and nothing really jumped out. So at this point, we realized how important it was to thoroughly and deeply identify what is the normal in-migration of bacteria in the gut. So we assembled 58 preterm infants, gestational age is 23 to 33 weeks. So that was a bit of a challenge because a 23-week gestational age child has a very different biology than a 33-week gestational age child. There's a gradient of risk of necrotizing enterocolitis, but this is a diverse population of subjects. To try to reduce some of the confusion, we focused only on one center at first, St. Louis Children's Hospitals. We sequenced all stools to day of life 30 from these children, and then every 3 days thereafter to day of life 60. This is the zone of risk of necrotizing enterocolitis. 922 samples, an average of 7,000 reads per sample. This is what we got. Now we take all the stools, 922 stools, 58 subjects, and we say, what bacteria are there overall? 20% bacilli. Bacilli, you would think would be gram-negative rods, but in fact they're gram-positive cocci. Gamma proteobacteria are the gram-negative rods, and Clostridia. There were multiple species within this, but the ribosomal database was able to crank out mostly bacteria we would all recognize. Now, if we gave everybody in this room a cup and everyone pooped and deposited it on the way out, and we sequenced the normal adult or older child population, it's a very different distribution. Gram-positives are there, but they are less common. Gamma proteobacteria, gram-negative bacteria, are actually rather low proportion in normal, healthy gut, and Clostridia are of higher proportion. Anaerobes are higher proportion. So that was the first rather high-level change we observed we had to deal with. Then we tried to look at interspecimen variability. This is another way to plot out data in multi-dimensions. The three taxa, Clostridia, Gamma proteobacteria, and bacilli, those three made up 93% of all the bacteria present in all the stool samples. And if you look at this plot and you look at the arrowheads, if your stool is a dot near a bacilli in the lower left-hand corner, that means your stool is largely bacilli. Lower left-hand corner will be gamma proteobacteria or gram negatives. And up in the upper right-hand corner, if you have a dot there, that means your stool is largely Clostridia, but it will be a blend of all. And there's not a tremendous amount of sense that can be made out of this. The color codes here are days of life. And if you really squint and you really imagine, you can say, well, there's a lot more blues towards the lower left-hand corner. These are the earliest children specimens. There's there's more polychromatic colors up towards the Clostridia, and it's still not very clear, at least at the level of the 922 samples that we put together. This is a single child who's actually rather prototypical, and most of the other single children we looked at did not have anywhere near as clear a progression as this child. So here are some of the challenges. Remember, we're trying to define the control group. Stools, even though they're a very safe specimen to handle, are produced when the child wants to produce them. You can't schedule a blood draw and say, okay, what is your weekly cholesterol level? So there's random stool days. The other challenge here is that there is a progression of bacteria. So earliest in life, a lot of blues here, a lot of bacilli. Next couple weeks, a lot of gram-negative bacteria. And this child, the organisms were largely E. coli, a subset of Klebsiella and Enterobacter. And then at around 30, 40 days of age, this child's stools began to start to become more abundant in anaerobes. So you see gram-positives, gram-negatives, anaerobes, a form of ecological succession, as David just described. So that sounds nice and clean, except there's a few little hiccups here. So second specimen, I think it's day of life 4, this child wants to all of a sudden knock down its gram-positives and found a few anaerobes to fill up the space and a lot of gram-negatives, but they didn't last. Several weeks later, this child tried to have a recovery of the bacilli. That too didn't last. So we have sample-to-sample instability, and you can begin to imagine you're going to need an awful lot of specimens to even begin to compare what's happening, especially in the context of, and that can occur any time along here. So what is normal early in life may not be quite so normal a week or two later. This is another way of looking at another subject, and you'll see these very sudden, abrupt changes in the population. Day-by-day samplings of a single child. Follow that arrow. You see the child started out having a lot of bacilli in the lower left, moved over to gamma proteobacteria, keeps going back and forth, and then inches its way towards Clostridia, and then stops with a stool that is not predominantly anaerobic, but equilibrates at about 40%. And all the children in this group converged on this consensus population at about the equivalent of 33 to 36 weeks post-menstrual age. So this is another way to look at this. You see bacilli predominant at first, Clostridia slowly rising in the aggregate, and gamma proteobacteria surging and then slowly being blunted. This is another way to portray our data. This slide, however, this was work from a mathematician affiliated with our statistician, Pato La Rosa, who demonstrates that the rate of rise of Clostridia is slower the more premature you are. So by 33 to 36 weeks, gestational post-menstrual age equivalent, your Clostridial population will be about the same if you don't develop necrotizing enterocolitis than it is in a child born more prematurely or less prematurely. Everything converges on the consensus, but the Clostridia get to that point quicker in children born less prematurely. It's the bottom row there. So when we put all this into a mixed model analysis, it was remarkable. While day of life predicted community structure and gestational age at birth predicted the rate of accrual of Clostridia, the communities assembled slowest in the most premature infants, and antibiotics, mode of delivery, feeds had little or no effect on the rate of this progression. So our interim conclusion, again, is we've really got to get a lot of stools to see if we're going to be able to associate necrotizing enterocolitis with a microbial population, and that statistics must account for multiple samples from the same individuals produced at unpredictable times. While we were gathering our specimens, 77,000 stools in total, several other manuscripts came out, and there were a variety of different organisms associated with neck risk. And Barb and I were, of course, concerned about being scooped, but our biggest concern was we would put together this massive collection and find no association, as some people were proposing. Nonetheless, we pushed forward. 77,000 stools in 3 centers: St. Louis Children's Hospital, Cosair Children's Hospital in Louisville, Kentucky, and the Children's Hospital at Oklahoma University in Oklahoma City, because we wanted to build in validation cohorts. We didn't want to repeat this study in series if we were to find anything, and fortunately, the NIH agreed with this. Controls were selected based on gestational age, weight, and closest in time in case there were temporal variations within an NICU. So I'll first show you, just to be clean, the data from St. Louis, cases versus controls. And you can see, look at the top panel there. On the left, there's more red prior to the development of necrotizing enterocolitis. Those are the cases. You can also see that there's more green and orange in the controls. And what we had formerly called Clostridia are now, in fact, Clostridia and negativicuides. There was a change in the ribosomal database version that we used, and that's why there's now 2 different classes of anaerobes. And you can see that there is a predominance of gram-negatives prior to the development of necrotizing enterocolitis. All these specimens were obtained before the event. The effect was strongest in the lower left-hand panel for children born at gestational age less than or equal to 27 weeks. We did not see such a strong effect in children born later. Now, fortunately, at least for the findings and the value of the findings, children born less than 27 weeks have the highest rates of necrotizing enterocolitis and the worst outcomes. In a mixed model analysis, of course, sick children will get neck and so on. We threw everything into the mix: antibiotics, route of delivery, maternal milk, again, small or inconsistent associations, and gamma proteobacteria. Presence of gamma proteobacteria was associated with neck, and presence of negativicuides, chiefly Veillonella, was associated with control status. This is the time by neck interaction, trying to take into account the dynamism of the progression of the population. We then brought in our 2 validation sites in Oklahoma City and Louisville. Combined, they had about as many subjects as we did in St. Louis. And when we put them into the mix, nothing lost significance. And in fact, one variable, the protective effect of negativicuides, did become significant in children over 27 weeks gestation at birth. So this is another component of observational studies. The first study is good, but the second study often puts you over the top, and the second cohort puts you over the top in making an association. And fortunately, we could not find any center effect. Now, gamma proteobacteria, you can see that the pie slices here, multiple different colors, meaning we could not define the risk beyond the level of gamma proteobacteria, gram-negative organisms. There may have been a slight center effect in which center had Klebsiella predominant or E. coli predominant, but the common denominator for gamma proteobacteria was gamma proteobacteria, and the LPS it would be bringing along. The common denominator for the negativicuides was one genus and species, Veillonella parvula. So once trying to reduce this to parsimony, we cannot get any more of a risk beyond gamma proteobacteria. We're trying to reduce this to protective parsimony. We could bring it down to a genus and a species level of Veillonella. Pictures of Veillonella at the bottom. Interestingly, Anthony van Leeuwenhoek is supposedly the first describer of Veillonella in a swab of oral, I don't know if he used a swab or it was a spit. When he looked at the microscope, it was probably Veillonella he was looking at from mouth flora. So again, and David and I did not coordinate our study, did not coordinate our talks. Is necrotizing enterocolitis the result of presence of bad? As physicians, as people trained in infectious diseases and microbiology, we like to think that germs can be bad, and we try to follow a pathogen paradigm. Gamma proteobacteria, certainly LPS-rich, gram-negative bacteria induce gut inflammation in piglets. Antibiotics can prevent that. There's a role in of TLR4 in work from Pittsburgh and elsewhere in experimental necrotizing enterocolitis development. The mouse models for this this disorder are not great. And this is the presence of bad model, where you have an abundance of gram-negative rods, the non-colored bars there, an abundance of TLR4 in the preterm gut, which is known to exist, and you have a match and a gas tank, and it suddenly results in necrotizing enterocolitis. But necrotizing enterocolitis could equally plausibly be the absence of good that in a population where anaerobes are relatively underrepresented, even with the right diet, you are not eliciting short-chain fatty acids, which are anti-inflammatory. So what about diversity? You cannot talk about the microbiome without at least mentioning the concept of diversity, and it's certainly an important ecological concept, but it's a bit challenging in this disorder. We have only 4 taxa that represent 93% of all the bacteria in the stool: gamma proteobacteria, bacilli, negative ichthys, and clostridia. So if one goes up and the other one goes down, that will automatically change your diversity. So it's very difficult to say with so few degrees of freedom, with only 4 taxa being present, that it is diversity per se as the cause of this, rather than a specific organism that is defining that diversity going up or down. So in conclusion, gamma proteobacteria negative ichthys defined dysbiosis, a different community structure associated with a bad outcome before NEC, something we think we've demonstrated, and I must say that others have suggested this in prior work. The effect is most apparent for infants born at less than or equal to 27 weeks gestational age. I can't answer the question, is this the cause of necrotizing enterocolitis, or is it only a biomarker? If it's only a biomarker, we can work with that. We got to prevent NEC. We got to do better than this. We cannot wait till a child has a bloody stool at 3 in the morning and the abdomen is distended to begin to address this problem. If we can identify this bacterial population, and those of you in the audience who are starting to think, I need a problem to bring my technology to, this is a fantastic one. We got to do better at predicting it, and predicting it may be that we will be looking at the microbial populations. Now, lest I leave you with the conclusion that all you need to do is take some stool, put it into a rapid sequencer, an hour later come up with the microbial content, I'd like to note that these curves here are all spline best highly confident averages. If you look very closely at the central tendencies here, there's not too many dots on those central tendencies. And the population, as I've showed you, is highly, highly unstable. It's kind of like the United States Census. The geographic center of the United States is in a small town called Hartville, Missouri. All the people in the West Coast, all the people on the East Coast. The geographic center where everybody lives is Hartville, Missouri. Nobody lives in Hartville, Missouri. So we may not be able to take a stool and say, you are at high risk of getting NEC, the way we've grown up considering your blood culture is positive. We get that. And in the end, we now have an opportunity for the first time to do microbiologically informed prevention of this bad coming in, or promotion of the good microbes or their products to prevent this horrible, horrible disorder. Again, thank you for listening, and I'd like to thank my team.